Welcome to chapter 19, where we will discuss viruses. A virus is an infectious particle consisting of genes packaged in a protein coat. A virus is a non-living thing. Viruses are much simpler in structure than even prokaryotic cells, and viruses cannot produce or carry out metabolism outside of a host cell. One phrase that can describe a virus is a borrowed life. Viruses exist in a shady area between life forms and chemicals. So again, viruses are non-living things. Viruses are not cells, nor are they made out of cells, and they also rely on cells in order to reproduce and survive. Here is a human immune cell infected with the HIV virus, the human immunodeficiency virus. And viruses were detected indirectly long before they were actually seen. The discovery of viruses. Tobacco mosaic disease stunts growth of tobacco plants and gives their leaves a mosaic coloration. In the late 1800s, researchers hypothesized that unusually small bacteria might be responsible. Later work suggested that the infectious agent did not share features with bacteria, such as the ability to grow on nutrient and media. In 1935, Wendell Stanley confirmed this latter hypothesis by crystallizing the infectious particle now known as tobacco mosaic virus, TMV. So here we see the experiment. He extracted the sap from the tobacco plant with tobacco mosaic disease and passed the sap through a filter that could trap the bacteria. He rubbed the filtered sap on the healthy tobacco plants elsewhere, and the healthy plants became infected. So there was an infectious particle that was able to go through the filter even when the bacteria were left behind. Again, viruses are not cells, therefore they are non-living things. A virus is a very small infectious particle consisting of a nucleic acid, either DNA or RNA, enclosed in a protein coat, and in some cases a membranous envelope. Viral genomes may consist of either double or single-stranded DNA or double or single-stranded RNA. Viruses are classified as either DNA viruses or RNA viruses. RNA viruses are also called retroviruses. The genome is either a single linear or circular molecule of the nucleic acid. Viruses have between 3 and 2,000 genes in their genome, so it's a very small amount of genes. A capsid is the protein shell that encloses the viral genome. Capsids are built from protein subunits called capsomeres. A capsid can have a variety of structures. Associated viruses may be referred to as helical or icosahedral viruses. So here we have different viral structures. First, we have the tobacco mosaic virus on the left, a adenovirus, influenza virus, and a bacteriophage, which is a virus that infects bacterial cells. Bacteriophages, also called phages, are viruses that infect bacterial cells. They have an elongated capsid head that encloses their DNA. A protein tail piece attaches the phage to the host and injects the phage DNA inside. Viruses are obligate intracellular parasites, which means they can replicate only within a host cell. And that means they can only reproduce by having a host cell. Each virus has a host range, a limited number of host cells that it can infect. The viral genome enters the host cell in a variety of ways. Once a viral genome has entered a cell, the cell begins to manufacture viral proteins. The virus makes use of the host enzymes, ribosomes, tRNAs, amino acids, ATP, and other molecules. So basically, once the virus injects its DNA into the host cell, the virus is going to use the host cell's machinery and materials in order to read the viral DNA and make parts of the virus in order to replicate more viruses. Viral nucleic acid molecules and capsomeres spontaneously self-assemble into new viruses. So here we see a virus infecting a cell, the host cell. The virus is going to enter the cell and uncode itself, releasing the capsid and also the genetic material, in this case DNA. The DNA can be further replicated, creating more viral DNA, or the DNA can be transcribed into an mRNA, which is translated to create more of the parts of the virus, which are the capsid proteins. The capsid proteins are going to self-assemble into the capsid protein coat, and also the viral DNA that was replicated are going to assemble as well into new viruses. The new copies of viruses are going to eventually exit the cell. So here we see a video of the viral reproductive cycle in a simplified version.
A virus is an intracellular parasite that can reproduce only by taking over a host cell. A virus consists of a nucleic acid genome enclosed in a protein shell called a capsid. In the virus shown here, the genome consists of DNA, but some viruses have RNA. Some viruses are also covered by a membranous envelope that is derived from the membrane of the host cell. There is usually a lock and key fit between the proteins of the capsid and receptors on a particular type of host cell. The virus attaches to a host cell and viral DNA enters the cell. The viral DNA uses nucleotides and enzymes of the host cell to replicate itself. The viral DNA then commandeers other host cell materials and machinery to transcribe its genes into messenger RNA and translate the RNA message into capsid proteins. Viral DNA and capsid proteins then assemble into new viruses. Mature viruses leave the host cell, often destroying the cell in the process. The viruses can go on to infect other cells, spreading the viral infection.
Phages, or bacteriophages, are the best understood of all viruses. Phages have two alternative reproductive mechanisms: the lytic cycle and the lysogenic cycle. The lytic cycle is a phage replicative cycle that culminates in the death of the host cell. The word lytic resembles the word lysis, which means to burst open. The lytic cycle produces new phages and lyses, or bursts open the host cell wall, releasing the progeny, or the children, of the viruses. A phage that reproduces only by the lytic cycle is called a virulent phage. So here is the lytic cycle at work. First, we have the bacteriophage virus infect a bacterial cell by attaching to it and then injecting its DNA into the bacterial cell. The virus is then going to use the cell's materials and machinery in order to read the viral DNA and make the parts of the virus. The parts of the virus are going to self-assemble, and therefore we create more of the progeny, or the children, of the virus. Once there are enough copies of viruses, they're going to exit the cell by bursting open the cell, a process also called lysis.
Bacteriophages, or phages, are viruses that infect bacteria. This is a T4 phage, which consists of DNA inside a protein coat. The lytic cycle begins when the tail fibers of the phage stick to receptor sites on the surface of a host bacterium, such as E. coli. The phage injects its DNA into the host cell, leaving the empty protein coat outside. The DNA of the host cell is destroyed, and host cell enzymes and nucleotides are commandeered to replicate the phage DNA, making more phage DNA. The host cell's enzymes and ribosomes transcribe the phage genes and translate them into phage proteins. Phage parts accumulate and assemble to form phages. A phage enzyme digests the bacterial cell wall and the cell ruptures, or lyses. As many as 200 phages spill out. Each of them may go on to infect another cell. This diagram summarizes the lytic cycle of bacteriophage T4.
The lysogenic cycle replicates the phage genome without destroying the host. So the cell is not going to burst open or go through the process of lysis. The viral DNA molecule is incorporated into the host cell's chromosome or genome. Phages that use both the lytic and lysogenic cycles are called temperate phages. The name of the DNA that's incorporated into the bacterial cell's genome or chromosome is called the prophage. Every time the host divides, it copies the phage DNA and passes the copies to the daughter cells or the progeny. An environmental signal can trigger the virus genome to exit the bacterial chromosome and switch to the lytic mode. So as you can see here, the lysogenic cycle is a lot more sneaky or insidious. We have the virus, the phage virus, inject the DNA into the bacterial host cell, and the viral DNA, instead of being read immediately, it's going to be incorporated into the bacterial DNA. The viral DNA that's incorporated into the bacterial DNA or genome is called the prophage.
In contrast to the lytic cycle, the lysogenic cycle reproduces the viral genetic material without destroying the host. The lysogenic cycle of phage lambda begins when a phage binds to the surface of a host bacterium. The phage injects its DNA into the host cell, leaving the empty protein coat outside. The viral DNA is incorporated into the host cell DNA, where it is called a prophage. Every time the host bacterium reproduces, it replicates the phage DNA along with its own and passes the copies on to daughter cells. Occasionally, the phage DNA exits the bacterial chromosome and initiates a lytic cycle. The viral DNA takes over the metabolic machinery of the host cell to make phage DNA and proteins. The host cell lyses, releasing phages which can infect other cells.
So we see here how a virus in the lysogenic cycle can go into the lytic cycle. We see the phage DNA in blue here, the virus DNA. And when it enters the lysogenic cycle, the viral DNA is incorporated into the bacterial DNA and becomes a prophage. The prophage is then further replicated when the bacterial cell reproduces and produces more daughter cells. And the progeny are going to have that prophage. However, that bacterial cell or the virus can go back into the lytic cycle. So there's going to be certain environmental triggers that's going to remove that prophage from the bacterial DNA, and it exits, and then the virus can go through the lytic cycle. And now the viral DNA right here is going to be read immediately in the lytic cycle, and the virus is going to use the materials and also the machinery, which are the enzymes and the ribosomes inside of the bacterial cell, in order to synthesize the protein parts of the virus. The parts are going to self-assemble, and we produce multiple copies of the virus. The phage virus then is going to eventually exit the cell, and when it does, it bursts it open, and this is called lysis. And that is why that is called the lytic cycle. So you can see how a virus in the lysogenic cycle eventually can come back into the lytic cycle and cause the death of the host cell. Bacteria have their own defenses against phages. Natural selection favors bacterial mutants with surface proteins that cannot be recognized as receptors by a particular type of phage. So when a viral DNA or a foreign DNA is injected into the bacterial cell, the bacterial cell has enzymes that can cut up the viral DNA. These are called restriction enzymes. The bacteria's own DNA is protected from the restriction enzymes by being methylated. Both bacteria and archaea, which are prokaryotes, can protect themselves from viral infections with the CRISPR-Cas system. It is based on sequences called clustered, regularly interspaced short palindromic repeats. All of that stands for CRISPR. Each spacer sequence between the repeats corresponds to DNA from a phage that had infected the cell. Particular nuclease proteins interact with the CRISPR region. These are called CRISPR-associated or Cas proteins. When a phage infects a bacterial cell that has the CRISPR-Cas system, the viral phage DNA is integrated between two repeat sequences. If the cell survives the infection, it can block any attempt of the same type of phage to reinfect it. The attempt of the phage to infect the cell triggers transcription of the CRISPR region. The resulting RNAs are cut into pieces and bound by Cas proteins. The Cas proteins use the phage-related RNA to target the invading phage DNA. The phage DNA is cut and destroyed. Natural selection favors phage mutants that can bind to altered cell surface receptors or that are resistant to enzymes. The relationship between phage and bacteria is in constant evolutionary flux. So here we see a diagram of the CRISPR-Cas system in a bacterial cell, which is a defense against the viral DNA that's injected by a bacteriophage virus. So the first step is, of course, the phage is going to inject its genetic material into the bacterial cell, and it triggers the transcription of the CRISPR region of the bacterial DNA. So right here is the bacterial DNA, and we can see that the bacterial DNA has DNA from previous infections by the same type of phage. The DNA sequences in between the DNA from previously infecting viral DNA are the repeats, the brown regions. Okay. So the infection is going to trigger the transcription of this DNA, the CRISPR region of DNA, into a RNA transcript, the mRNA. Step 2 is the RNA transcript is processed into short RNA strands. As we can see here, we have a short RNA strand. And then step 3, the short RNA strands are going to attach to the Cas protein here. So each short RNA strand binds to a Cas protein, forming a complex. And step number 4, the complementary RNA binds to DNA. The Cas protein cuts the phage DNA. So the short RNA piece is going to find its complementary DNA on the template strand of DNA, and at that specific location where it matches with the DNA, the Cas protein is going to cut that section of DNA. Because we cut that piece of DNA so the DNA is the viral DNA, the phage virus DNA can no longer replicate. So we have a defense mechanism against viral DNA in bacterial cells. Here's a computer model of the CRISPR-Cas9 gene editing complex from a bacteria called Streptococcus pyogenes. There are two key variables used to classify viruses that infect animals: an RNA or DNA genome, either single-stranded or double-stranded, the presence or absence of a membranous envelope. The big difference is that virus a DNA or a RNA virus? Whereas few bacteriophages have an envelope or an RNA genome, many animal viruses have both. So most viruses that infect bacteria, also called bacteriophages, have DNA as its genome. Many viruses that infect animals have a membranous envelope. Viral glycoproteins on the envelope bind to specific receptor molecules on the surface of a host cell. The viral envelope is usually derived from the host cell's plasma membrane as the viral capsids exit. Here we see an example of an RNA virus with an envelope that's going to infect the host cell. And so unlike the bacteriophage, because this virus has an envelope, it's going to actually fuse into the host cell by fusing with the host cell's cell membrane. Once it enters the host cell, the virus is going to disassemble and basically release its genetic material into the host cell. The genetic material in this case is going to be RNA, and the mRNA can either be translated into different capsid proteins, which can be used to assemble the new copies of viruses, or the mRNA can be further copied into more mRNA or RNA, and that RNA can be put into the capsid coat as part of the process of making new copies of the RNA virus. And also, as you can see, the glycoproteins from the endoplasmic reticulum are going to be used. So the host cell's glycoproteins are going to be used to attach to the membrane or the envelope of the copies of the new RNA viruses. Other viral membranes form from the host nuclear envelope and are then replaced by an envelope made from Golgi apparatus membrane. The broadest variety of RNA genomes is found in viruses that infect animals. RNA viruses are also called retroviruses, which also use an enzyme called reverse transcriptase to copy their RNA genome into DNA. So just like how the name of the enzyme sounds like, it describes exactly what it does. So reverse transcriptase helps to catalyze reverse transcription, which is going from RNA to DNA. An example of a retrovirus is HIV, human immunodeficiency virus, which is a retrovirus that causes AIDS, acquired immunodeficiency syndrome. The viral DNA that is integrated into the host genome is called a provirus. Unlike a prophage, a provirus remains a permanent resident of the host cell. RNA polymerase transcribes the proviral DNA into RNA molecules. The RNA molecules function both as mRNA for synthesis of viral proteins and as genomes for new virus particles released from the cell. So here we see several classes of animal viruses. We have double-stranded animal viruses. We have single-stranded animal viruses, and they can have either DNA or RNA as their genetic material. And as you can see in the charts, it also shows you whether they have an envelope or not. So here we see a diagram of HIV infecting a virus. And as you can see, HIV has an envelope. So HIV, it's going to fuse with the host cell. But first of all, we need to know that HIV has a certain host range. In this case, HIV is infecting a white blood cell, and it recognizes the host cell, which is the white blood cell, by attaching to receptors located on the cell membrane of the host cell with its glycoproteins located on its envelope. So once it does attach to the receptors of the host cell, the HIV virus is able to fuse into the cell and eventually release its RNA genetic material. Along with the RNA, the HIV is going to release the reverse transcriptase enzyme, which is going to convert the RNA into DNA. The DNA is going to be incorporated into the genome of the host cell, and once it's incorporated into the genome of the host cell, it's going to be called the provirus. The provirus is then going to be transcripted into a mRNA, or transcribed into an mRNA. The mRNA can either be used to put into the new copies of the HIV virus, or the mRNA can be used to translate it into the different parts of the protein parts of the HIV virus, such as the capsid, or making the glycoproteins. And as you can see, we make new copies of the HIV virus, and they're going to eventually exit the cell by fusing out. And remember from previous chapters when, so this is kind of similar to endocytosis in a sense, and when the HIV virus exits, it's similar to exocytosis. So it kind of looks similar to that. So here we see an electron micrograph of the HIV virus infecting a white blood cell. And let's take a look at the reproductive cycle of HIV.
Understanding the structure and life cycle of HIV, the AIDS virus, is critically important to dealing with the disease. HIV is covered by an envelope derived from a host cell membrane. Glycoproteins studding the envelope recognize and bind to receptor molecules on the host cell. A protein coat surrounds the viral genetic material, which consists of 2 molecules of single-stranded RNA. The enzyme reverse transcriptase enables the virus to make DNA from an RNA template, a trick for which this group of viruses is named retroviruses. The RNA molecules of HIV enter a host cell when the virus fuses with the plasma membrane and the coat proteins are removed by enzymes. Reverse transcriptase catalyzes the synthesis of a DNA strand complementary to the viral RNA strand, and then a second DNA strand complementary to the first. The double-stranded DNA is incorporated as a provirus into the host cell's chromosomal DNA, where it may lie dormant for years. Occasionally, the provirus is transcribed into RNA. This RNA serves as both messenger RNA for the formation of HIV proteins and as genetic material for the next generation of viruses. Protein coats form around viral RNA and reverse transcriptase molecules. Viruses bud from the host cell, acquiring envelopes as they leave.
Viruses do not fit our definition of living organisms. Viruses need a host cell in order to reproduce. And also, viruses are not cells, nor are they made of cells. This is why viruses are considered non-living. Candidates for the source of viral genomes include plasmids and transposons. Plasmids, transposons, and viruses are all mobile genetic elements. The largest virus yet discovered is the size of a small bacterium. Its genome encodes proteins involved in translation, DNA repair, protein folding, and polysaccharide synthesis. There is controversy about whether this virus evolved before or after cells. Diseases caused by viral infections affect humans, agricultural crops, and livestock worldwide. Smaller, less complex entities called prions also cause disease in plants and animals respectively. Viruses may damage or kill cells by causing the release of hydrolytic enzymes from lysosomes. Some viruses cause infected cells to produce toxins that lead to disease symptoms. Others have molecular components, such as envelope proteins, that are toxic. A vaccine is a harmless derivative of pathogenic microbes that stimulate the immune system to mount defenses against the harmful pathogen. Vaccines can prevent certain viral illnesses. Viral infections cannot be treated by antibiotics. Antibiotics are only going to be used to treat against bacterial infections, not viral infections. Antiviral drugs, however, can help to treat but not cure viral infections by inhibiting synthesis of viral DNA and by interfering with viral assembly, which was putting the different protein parts of viruses together to make copies of the new viruses. Emerging viruses are those that suddenly become apparent. The Ebola virus is one of several emerging viruses that cause hemorrhagic fever and often fatal illness. Other examples include the chikungunya virus and the recently emerging Zika virus in 2015. So here are micrographs of the Ebola virus, the chikungunya viruses, and the Zika virus. In 2009, a general outbreak called an epidemic of a flu-like illness appeared in Mexico and the US, caused by an influenza virus named H1N1. A global epidemic is called a pandemic. Three processes contribute to the emergence of new viral diseases. RNA viruses have an unusually high rate of mutation. The disease can be disseminated from a small, isolated human population and can eventually spread around the world. About 3/4 of new human diseases originate by spreading to humans from animals. Flu epidemics are caused by type A influenza viruses. These infect a wide variety of animals, including birds, pigs, horses, and humans. Strains of influenza A are given standardized names based on the viral surface proteins hemagglutinin, HA, and neuraminidase, NA. H1N1 is the strain that caused the 2009 flu pandemic. Changes in host behavior or the environment can increase the spread of viruses responsible for emerging diseases. New roads into a remote area may increase spread of viral diseases. The use of insecticides and mosquito nets may help prevent the spread. It is possible that global climate change may allow mosquitoes that carry viruses to expand their range. So as we can see here, mosquitoes are great vectors or carriers for disease. And there are certain ways to prevent transmitting diseases, possible diseases, from getting bitten by mosquitoes. And here's a net protecting this child. Plant viruses spread disease by two major routes: horizontal transmission, entering through damaged cell walls, or vertical transmission, inheriting the virus from a parent. Prions are infectious proteins that appear to cause degenerative brain diseases in animals. Prions are incorrectly folded proteins which can be transmitted through food. They act slowly and are virtually indestructible. Scrapie in sheep, mad cow disease, and in humans are all caused by prions. Creutzfeldt-Jakob disease is another example. Prions are somehow able to convert a normal form of the protein into the misfolded version. Then several prions aggregate or stick together into a complex that can convert more proteins to prions. Prions might also be involved in diseases such as Alzheimer's and Parkinson's disease. So here we have a misfolded protein called a prion, and it's going to be highly contagious. The prion is going to bump into a normal protein and change that normal protein into a misfolded protein. The new prion is going to aggregate or combine with the original prion, and over time all the normal proteins are going to turn into the misfolded prion proteins. This is going to cause devastating neurological effects.